Kimchi is a traditional Korean side dish of salted and fermented vegetables such as salted cabbage mixed with radish, green onion, red pepper powder, garlic, ginger, and fermented seafood. Kimchi is made by lacto-fermentation of cabbage, which means the fermentation by lactobacillus bacteria. The red pepper used in fermentation of kimchi contains capsaicin, which is an active compound in chili peppers. They are plants that belong to the genus Capsicum. Capsaicin has multiple pharmacological and physiological properties. For example, it is used in pepper spray, which is a self-defense object to cause pain and tears in the eyes, with the active ingredients being oleoresin capsicum, which is extracted from chili peppers. Capsaicin has also been reported to downregulate the expression of prostate-specific antigen androgenic receptors. It kills the prostate cancer cells by causing them to undergo apoptosis. Capsaicin has also received so much attention because of its antimicrobial and antiviral activities. Studies have shown that capsaicin exerts a growth inhibitory effect on bacteria. The purpose of this study was to investigate how the different levels of red pepper powder, from mild to extreme, exhibits a growth inhibitory effect on bacteria present during kimchi fermentation. So the first objective of this project was to establish kimchi cultures containing various concentrations of red pepper powder. Then after, investigate their pH, organic acids, lactic acid bacteria count of the samples collected at various time points. And finally, determine their microbial diversity present at various time points using RT-qPCR. So the first step was to prepare and ferment the kimchi. So the kimchi samples were made with the same concentrations of cabbage, carrot, sugar, ginger, garlic, and fermented anchovy sauce, as well as water, and different levels of red pepper powder were used. The samples were made in biological duplicates and then left in the fridge to ferment for 37 weeks at -8 degrees Celsius. So the cabbage, carrot, sugar, ginger, garlic, they were all weighed on a scale. So the same concentrations of all of the ingredients were used for the preparation of the kimchi. So the next step was to do the sampling and the pH analysis of the kimchi samples collected at the first time point. So the kimchi samples were homogenized and placed in their respective jars. Then the samples were collected at 7, 17, 26, and 37 days of fermentation, and they were filtered using a cheesecloth, after which a pH meter was used to check their respective pH, and that was recorded. And after that, for the same samples, the lactic acid bacteria count was determined using 3M petrofilms. So about 1 milliliter of the filtered samples were diluted 10 times with 0.1% peptone water. Then 1 ml of the diluted sample was dispensed right at the center of the bottom film of the 3M petrofilm plate, and the plate was then incubated at 37 degrees Celsius for 48 hours. Then after that, the organic acid content of the samples were also determined using UPLC. So the filtered samples were obtained, and then 1 ml was taken and centrifuged at full speed for 10 minutes. After which the supernatant was taken out into a new tube. So after that, the samples were diluted 1x with UPLC grade water and then filtered into UPLC vials. The standards of the run were tannic acid, citric acid, lactic acid, and malic acid. Then after that, the DNA was extracted from the samples and amplified using RT-qPCR. So the DNA extraction was done just for the first sampling and the final sampling to compare the bacterial diversity between the first day of fermentation and the last day of fermentation. So the DNA was extracted from the samples using the GenElute bacterial genomic DNA kit, and the concentrations and purity of the extracted DNA was checked on the nanodrop spectrophotometer. An aniline temperature and primer efficiency run was then carried out for all of the primers, and the PR applicants were run on a 1% agarose gel. The extracted DNA of the samples collected on the 7th and 37th day of fermentations were then amplified using RT-qPCR, and 16S rRNA was used as a housekeeping gene. Results. So the first figure, and that's figure 1, shows the pH comparison of the control and the treated samples at different time points. So as seen from the image, the pH of the samples were in the range of 4.5 to 5 at the 7th day of fermentation. Then they all decreased relatively at the same rate throughout the rest of the fermentation process. So that means that there was no difference of the different levels of red pepper powder and the pH of the kimchi samples that were fermented. So the second figure there, and that's figure 2, shows the lactic acid bacteria enumeration of the kimchi sample treated with 25 grams of red pepper powder at the beginning of the fermentation and at the end of fermentation. The number of colonies present at the beginning of the fermentation process was approximately 1,455 colony forming units, while the collected on the 37th day of fermentation had 28 colony forming units. And the third image, and that's figure 3, shows the lactic acid bacteria enumeration of the kimchi sample treated with 0 gram of red pepper powder at the beginning of fermentation and at the end of the fermentation process. So both of the plates had they produced heterofermentative colonies, where the one for the 37th day of fermentation produced a higher concentration of heterofermentative colonies. There was little to no growth gotten for the kimchi fermented with 0 gram of red pepper powder, and that's the negative control. Results that is here is the one gotten from the UPLC. So figure 4 shows the chromatogram of the final sampling treated with 25 grams of red pepper powder, and the one right beside it is the one treated with 0 gram of red pepper powder. So from the UPLC results, it showed that all of the samples had about the same content of organic acid in them, with tannic acid being having the highest concentration in all of the samples. So figure 5 shows the primer efficiency run for the two of the primers used, and that is L. gelidum and W. candleri. So the PCR samples were diluted from neat to 10 to 6, and all the primers used has an efficiency between 90 to 110. And the final image here, and that's figure 6, shows the statistical analysis of the first and final sampling. So there was more bacteria present in the kimchi samples from the first sampling compared to the final sampling. And there was no significant difference in the bacterial diversities present in the samples treated with different levels of red pepper powder. So going to the discussion and conclusion. The pH profiles of all of the kimchi samples decreased rapidly after the first week of fermentation, and then became approximately the same in the range of 1.0 and 3.6. The pH profiles for all the kimchi samples were similar, regardless of the addition of red pepper powder. The lactic acid bacteria count of the kimchi sample significantly decreased over time. This should not be the case, as more lactic acid bacteria are being produced as the fermentation progresses, but a reasonable explanation for this would be, since the kimchi samples were all homogenized after the first sampling, then there was no whole kimchi or no whole ingredient for the bacteria to feed on. Another explanation could be the conditions the samples were left to ferment in, and the kimchi samples were placed in the -8 degrees fridge, which could have slowed down the fermentation of the kimchi samples. The organic acid content analysis showed that the same organic acids were present in the samples with approximately the same concentrations, regardless of the addition of red pepper powder. Finally, there was no significant difference in the bacterial diversity within the treatment groups from the first and final sampling. However, there was a notable difference in the bacterial diversity between the first sampling and the final sampling, with the final kimchi sample collected having less diversity. So in conclusion, further studies need to be carried out on this topic without homogenizing the whole kimchi and fermenting the kimchi at room temperature so as to get a clearer understanding on what effects a higher concentration of red pepper powder has on kimchi fermentation. Thank you.